Imagine a world where vaccines come not in vials and needles, but in fruits and vegetables. Welcome to the revolutionary concept of edible vaccines, where genetic engineering meets immunology to create a new paradigm in global health. Edible vaccines are produced in genetically engineered plants that express specific antigens. These plants are modified to produce immune-stimulating proteins from pathogens, and can be simply eaten to stimulate the body's immune response against diseases. In 1990, Charles Arntzen introduced the concept of using transgenic plants as vaccine production systems. The first successful expression of a bacterial antigen in tobacco plants marked a breakthrough in 1992. By 1998, scientists achieved expression of hepatitis B surface antigen in potatoes, proving clinical potential. Transgenic plants produce pathogen antigens in their cells through genetic engineering. These antigens are consumed and partially digested, interacting with gut-associated lymphoid tissue to initiate an immune response. Both mucosal and systemic immune responses develop, providing protection against diseases. Edible vaccines are cost-effective, requiring only sunlight and basic nutrients. They are needle-free, reducing pain and medical waste. Many plant tissues preserve antigens at room temperature, and they trigger immunity at pathogen entry sites in the digestive tract, enhancing protective effects. Developing edible vaccines involves overcoming dosage standardization, complex regulatory hurdles, public acceptance, and allergenicity concerns. These challenges form a complex web of obstacles that scientists and regulatory bodies must navigate to bring edible vaccines from concept to reality. Fruits like bananas, tomatoes, and papaya offer child-friendly delivery systems. Vegetables such as potatoes, carrots, and lettuce provide excellent antigen expression platforms. Grains like rice, corn, and wheat offer stability and storage advantages. Potato tubers allow for exceptional antigen accumulation and preservation. Well-established genetic modification protocols make potatoes ideal for research. Antigens in potato tissue remain viable for months at room temperature, though heat from cooking can denature vaccine proteins. Bananas are ideal for pediatric vaccination due to their sweet taste and soft texture. They grow naturally in many developing regions with high disease burdens, reducing transportation costs and import dependencies. However, their slow development cycle presents challenges for rapid vaccine production. Microalgae represent an emerging frontier in edible vaccine development. They complete their life cycle in days, require minimal resources, and produce abundant antigens. The Model C Reinhardt T offers a proven expression system, making algae particularly promising for rapid response vaccine production scenarios. Various techniques are used to introduce foreign DNA into plant genomes, including agrobacterium-mediated transformation, biolistic methods, electroporation, and viral vectors. These methods enable the creation of transgenic plants that express vaccine antigens efficiently. Edible vaccines are being developed for diseases such as cholera, hepatitis B, malaria, and COVID-19 using plant platforms like rice, potato, lettuce, spinach, and tobacco. These vaccines are in various stages of development, from preclinical to early research. Researchers successfully expressed Norwalk virus capsid protein in tobacco and potato plants. Volunteers who consumed transgenic potatoes developed specific antibodies against Norwalk virus. This milestone study provided the first human proof of concept for edible plant-based vaccines. Veterinary applications represent the fastest path to market for edible vaccines. Livestock vaccination faces fewer regulatory hurdles and addresses significant economic challenges in animal husbandry, making it a promising area for edible vaccine development. Plants must meet strict genetic modification regulations that vary by country and region. Good manufacturing practices must be adapted for agricultural production environments. Methods must ensure uniform antigen expression across different growing conditions, and safety and efficacy trials must meet both pharmaceutical and agricultural standards. Novel proteins must be assessed for potential allergenic reactions in consumers. Plant-specific sugar modifications can improve immune responses and safety. Strategies are needed to prevent accidental consumption by non-target individuals, and containment measures are required to prevent cross-pollination with food crops. Ensuring consistent dosage remains the greatest challenge in edible vaccine development. Environmental factors can cause antigen expression to vary significantly between individual plants, making standardization and consistent delivery critical for effective immunization.